Hello everyone. So in this video we are going to talk about another two topics of developmental biology about self-fate and cell commitment. Right. So here is self-fate. Let's start with its definition. So self-fate. Self-fate describes the range of cell types a particular cell can give rise to during normal development. So, in similar words or in easy words, what it means is that all the kinds of cells that can form from a cell could be sulfate, right? So, for example, if this is a blastomere and these are the cells, blastomeres is the embryonic stage where there are 60 to 120 cells. So blastomere stage, and some of these cells will form the muscles, some of these will form the notochord, some of these will form the the epidermis, right? So all these will have different kind of fate, right? All these cells will have of a blastula of an embryo, developing embryo, will have different kind of cell fates. So that is cell fate, where there is all the range of cell types a particular cell can give rise to during normal development. So different, they have different cell fate. So now it's about commitment. So commitment is the deciding of the fate. So commitment simply means when a cell had decided what its cell fate will be, that it will, when a cell will differentiate and form a definite kind of cell, a definite kind of tissue, what will it form? If that had been decided, that process of deciding is known as commitment. That cell is committed. That would mean that cell had decided what it is going to become. So there are two stages of commitment: specification and determination, right? So let's see this in a clear view. If this is a blastomere stage of the developing embryo, this is a blastomere with many cells of a developing embryo. So if this cell had decided to form the ectoderm, if its fate had been decided to form the ectoderm, then this cell is committed. Committed cell. But if this cell can either form the muscle or could form the the gut region. So if it is not decided what's going to be formed, it's not committed, right? It's a non-committed cell. So there are many kind of commitment, the specification and determination. This is temporary commitment. The specification or determination is the permanent commitment, where the fate could not change. Once decided, once determined, the fate could not change of that cell, couldn't change in determination. But in case of specification, the fate could change. The fate could change, right? So that's about these two types of commitment. The specification is a temporary kind of commitment, the determination is a permanent kind of commitment. In this determination, when the cell has been determined, when it is decided that I am going to differentiate to become this, there is no changing that. It's something like students, when we are in primary school, we decide, decide. One day we think we are going to become pilot, the other day we think, oh, let's become something else, a teacher, something else, a doctor. So that's kind of commitment. One day something, the next day something else. So that kind of commitment, the temporary commitment, is specification of cells. But in case of determination is when, right now, if you are watching this video, you are probably uh, preparing for your UGC net preparation or your BSc preparation. So you already determined what you are going to do. So that's kind of determination, right? Specification. Now, what is specification? It is a liable phase, so it is not fixed. It can revert from its fate. So, in this case, commitment is reversible. Like I said earlier, there are two types of commitment, the specification and determination. Specification, case of specification, it is a liable phase, that is temporary phase, so not fixed. So right, so for example, if a cell, if this is an embryo or a blastomere, and these are the cells, and if this cell is specified to become muscle, right, it is specified to become muscle, and we cut it out, the cell, we take this cell out which is specified to become a muscle cell, we take this cell out and put it into another embryo, right, we put it here somewhere where these cells are going to become the gut region, right. We take out a cell that is committed to become muscle and we put it into the gut region. So now the cell would not no longer form muscle but will form gut, right. So that kind of commitment is specification, that kind of commitment which could be changed, that is specification, right. So it can revert from its fate. In this case, the cell could revert from its phase and commitment is reversible. Let's go to the next slide. Determination, of course, this is the second kind of commitment, determination, where once decided it could not be reversed. So in determination, commitment is fixed, so it is not going to revert from its fate, right. So in this case, commitment is irreversible. So if a cell is determined, it can differentiate to form only one kind of cell. That's easy to remember, if this is again a blastomere with cells, and these are the cells, and if this cell is supposed to become muscle, right, this it has been determined to become muscle, right. So what would happen if you take this cell out and put into another embryo in the place of gut cell, right, or nephron cells to form kidney, right. So this cell, you put this cell, take out the muscle cell and put it into a developing embryo uh, among the kidney forming cells, but you will see that this cell, if it is determined, would still form muscle, it would still form a muscle and not a kidney. So it is determined cell, determination had occurred, so the fate of the cell could no longer be changed, right. You could not change the cell's mind now, so it would only form what it had decided to become. Now, differentiation. Cell differentiation is the process by which the cell becomes structurally and functionally specialized, allowing the formation of distinct cell types. Now, that is easy to remember. We already know what differentiation is. It is when there is zygote 2n, there is zygote single cell, it cleaves to form two cell stage, then four cell stage, then eight cell stage, right. Eight cell stage to something 120, and then more and more to a multicellular organism is formed, right. A multicellular organism, complex organism is formed like this from a single cell. So when that is formed, these cells differentiate and specialize to form a different. Now we all know that our life had started from a single cell zygote, right, but now we have all kind of cells in our body, from nephrons in kidney to neurons in our brain, nephrons in kidney, neurons, our skin cell which is the epidermis, and our backbones, bones. We have osteocytes, we have all kind of cells, the red blood cells. So we have all kind of cells in our body, while our life had started from a single cell, the zygote. So the cell had divided and then differentiated, right, had become structurally and functionally specialized to form special functions, right. Each cell had become specialized. So that process is known as differentiation, right. When the cells become specialized structurally and functionally, right, like a nephron has some kind of like this structure, and this is cell body and its dendrites and axon. So it is structurally and functionally specialized to send signals from one cell to the other, while a red blood cell, something like this red blood cell, the RBCs, are functionally and structurally prepared as differentiated so that they could carry oxygen, right? So you can carry oxygen. So this is differentiation, where the cells become structurally and functionally specialized, allowing the formation of distinct cell types. So that's differentiation. Thank you for watching. Please like and subscribe the channel.